Sorry about that. So as I mentioned this morning in the opening comments, we really structured the afternoon sessions to look forward to the future. And I'm particularly excited about this session and so grateful to all the panelists, but in particular our chair, Dr. Dolsten. So the promise of gene therapy that Dr. Dolsten put together is a panel of experts that can really help us both dream about the future, but in so many ways the future is now. And so I think it helps us put into context why some of the promise and the peril of innovation that we heard about this morning is so very important for us to sort out, because this is one of the ways that we can use what, for people like me who've been in this for a long time, think of as a magic word, which is cure. And so with that promise and peril, take it away.
Thank you very much, Sue, for that introduction. It feels like a special moment in time here, having followed and at various time points touched gene therapy over a few decades. And at this very time point, we have gathered a very esteemed panel that will share with you some of the most remarkable medical advances that are happening right now, from recently approved drugs to clinical development and regulatory filing that is to change completely medical practice and provide hope to patients and families that probably never thought they would experience such transformative treatment opportunities. So I'm very pleased to have here on my left Katherine A from Spark Therapeutics. And Kathy is a pioneer in the field of gene therapy, originally starting and advancing the field at UPenn and with others building Spark Therapeutics. That was the first company with the support, obviously, from the division at the FDA to get the first approved drug for gene therapy in the United States, Luxturna, for hereditary eye disease approved. I've also had a privilege to work with Kathy and her talented team at Spark on the Factor 9 gene therapy project that we are partnering on and advancing now together. Next, we have Peter Marks from the CBER division of FDA that manages these complex therapies from a regulatory point of view. And Peter is also a champion of providing regulatory policies and guidance that allow this field to really overcome obstacles and advance in a consistent and controlled manner these remarkable technologies that are now knocking on the door of cure, as we heard in Sue's introductory words. And I think what's also very important is the role of Peter and FDA to try and harmonize regulations worldwide to make sure we can accelerate, whenever successful, these treatments to patients in urgent needs across many different regions. Next to Peter, we have Brian Caspar, another academic pioneer that over a few years have done the remarkable journey from Nationwide Children's Hospital, where Brian was one of the faculty members pursuing rare neurological disease with a particular interest for SMA. As you know, one of those fatal conditions where kids are diagnosed in the early months of life with a difficult neuromuscular disease that usually, unfortunately, lead to fatal outcome around 1.5 years. And through the discoveries in Brian's lab that led to the foundation of AveXis, remarkable gene therapy data has been clinically noted, and I think you're now in registration phase and have done the personal journey from academia, biotech to big pharma. And we would certainly enjoy listening how you've crossed those exciting but sometimes, of course, demanding boundaries that allow you to do different things in different ways. Then next to Brian, we have Gregory from Bluebird that, in contrast to Brian and Kathy that have worked on in vivo single infusion gene therapy with AAV vectors. Phil represent the use of lentivirus and ex vivo transfection of selected blood cells to provide improved function to patients with blood diseases such as thalassemia and severe sickle cell disease. And I know, Phil, with this novel technology, also at an exciting time point in a regulatory phase, I think you actually are first pursuing the European path, and we look forward very much to learn how this procedure may change completely the lives of these patients. And last but not least, Gaurav Shah, that also uses in the initial efforts of Rocket Pharma, where Gaurav is one of the founders, uses also lentivirus for another difficult disease, Fanconi anemia, representing again this approach to ex vivo transfer deficient genes and to restore functions of blood-derived cells. And I think we'll really enjoy this New York-based biotech that is neighbor with us here in the next building. You know, what's really common between the members of this panel is that they have embarked on either regulating or discovering and developing breakthrough technologies in diseases which are genetically defined, but were kind of beyond the reach for medical intervention. And we see right in front of us on conferences or high-profile papers in New England Journal of Medicine, how these data really in a remarkable way seem to provide unprecedented efficacy for fatal diseases. And it's an exciting journey that have started, of course, propelled by the understanding of the human genome some years ago and the ability to get deeper insights into some of these hereditary diseases. And for Pfizer and myself, it has been an exciting last 3 years or so as we joined this team of gene therapy entrepreneurs, both internally and by collaborating with many of these biotech groups in kind of a networked R&D model, including, as I mentioned, on hemophilia type B with Factor IX collaboration with Katherine A, and Spark, where we're on the path to starting pivotal study after also reporting dramatic effects in these hemophilia patients in restoring the factor function and basically abrogating the need for any continuous infusion as seen in the currently running clinical studies for a couple of years. We then had opportunity to continue to collaborate with groups such as Sangamo. I actually think, Phil, you had a previous experience with that company where we work on Factor VIII, and I know Kath is also involved in that field, trying to expand into other hemophilia diseases. We had an opportunity then to collaborate with spin-outs from University of North Carolina, the founder Jude Samalski with Bluebird Therapeutics that we acquired in order to get into neuromuscular disease. And that area around North Carolina, Duke, and UNC is another big center for gene therapy, where we are very pleased to be heavily present and not just pursuing science, but producing clinical material and also manufacturing. That latter piece is so important as we see these dramatic advances to be able to plan for success as you see promising clinical data and make the important improvement to take experimental early clinical phases into planning for potential approvals and providing sustained, reliable, effective manufacturing for patients worldwide. So during this journey, I think a number of questions that arise to all of us here is, on one hand, how we as a medical and scientific community can continue to advance insights about hereditary diseases to be able to address additional patient groups, how we can apply our knowledge and grow our knowledge around these delivery systems, whether AAV or lentivirus, How do we make them more efficient in transfecting the cells that we're aiming for and not the unwanted cells? How do we increase the persistence or the production level of the transgenes we want to deliver? And how do we make manufacturing more effective or more potent materials that we can deliver lower dose and think about being able to provide increased access to patients across the globe, not just in the most developed part of the world? And to make all of that happen, we need to have regulatory standards that are consistent across regions and that allow us to learn from each product area and to be able to get guidance that allow us to advance products in a safe but accelerated manner in order to face and some of the great unmet medical needs, but do it with urgency to be able to serve these patients. And I think we'll hear all of these aspects during the exciting period of this panel. So with that introduction, I would like to have each of the panelists share some of their experiences in a few minutes. Then I will return with a pointed question to each of them to warm up. And meanwhile, all of you, think about what's the question you always wanted to ask to one of the most prominent scientists in the world in gene therapy, and you can direct it to anyone present here. This is your chance to learn, to ask questions, and to advance the field by constructive, challenging ideas. Kathy, please.
Okay. Michael, thank you for that rousing introduction. So my name is Kathy A, and I am a co-founder, president, and head of R&D at Spark Therapeutics. Spark was founded 5 years ago, and our company is really devoted to discovering, developing, and delivering gene therapies for genetic disease. The history of our company and its founding is really closely intertwined with the history of clinical gene therapy, which began in 1990 at the NIH in a trial for ADA-SCID. The history of clinical gene therapy had a quiet childhood and a difficult adolescence. After that quiet childhood and the beginning of the difficult adolescence, the partner that we had been working with, a company in Northern California, turned away from gene therapy, as did many of the early companies from the 1990s, and we found that we had to set up work internally at Children's Hospital of Philadelphia to manufacture clinical-grade vector, to do our own regulatory work, because most companies were turning away from this space. Our programs went well. One for a rare inherited retinal dystrophy. You heard Michael talk about Luxturna, which was approved last year by the FDA and which has recently received a positive opinion from the CHMP of the EMA. That was approaching phase 3. We had to determine how we were going to move it through phase 3 and onto approval. We felt that we could not do that at Children's Hospital of Philadelphia. We considered many options, and we eventually determined to set up a company that was spun out from our group at Children's Hospital that took over the programs that were ongoing there in hemophilia B, as well as in this rare form of inherited retinal dystrophy, and began the company. The company has now expanded. It's located in Philadelphia, about 8 blocks away from Children's Hospital of Philadelphia, and now has 350 employees and is working on programs across a number of target tissues, the retina, the liver, and the central nervous system. Our 2 lead programs, as Michael indicated, Luxturna has already been approved. Hemophilia B, now in phase 3, sponsored by Pfizer, represented 2 very different problem sets in gene therapy. And if we have time, we can talk about the differences there. For Luxturna, one of the most important problems was that for a class of diseases that has not previously had therapeutics, it became important to actually develop and then validate a novel primary endpoint. We had a lot of collaboration with the FDA, Office of Tissue and Advanced Therapies, to get that done. And hemophilia represents the problem of how do you understand and manage the human immune response to a recombinant virion that's been engineered from a virus.
So thank you. So I'm going to give you a perspective of FDA, where we see, obviously, there is one directly administered and 2 cell-based gene therapies that have been approved. But we see this in the context of well over 700 investigational new drug applications in the gene therapy field. So there's a lot of work going on here. And we see that as, and I would see that as something, clearly we're at a potentially transformative period in medicine where gene therapy has the potential to address any number of rare diseases. But I think we should start to think larger than that, because besides hemophilia, retinal dystrophies, it also potentially has the ability to actually address some more common diseases. But there are some things that are going to have to happen if we're going to get there, and I'll say more about that in a minute. What we're doing at FDA to try to help move things ahead from a regulatory perspective is use our expedited pathways, expedited programs such as Breakthrough Therapy Designation, Regenerative Medicine Advanced Therapy Designation. That is to try to give these products the most agency input, senior leadership input from the agency, use of the expedited programs such as priority review, which gets things a 4-month faster review, use of our accelerated approval provisions, so that these things have the best chance of seeing the light of day. We've also started to try to de-risk some of the development, particularly for smaller companies that are developing things, by allowing for earlier non-binding regulatory meetings as part of something that we've named it the Interact program, where basically academics or generally smaller companies will come in and have a meeting with us that's in an informal basis to ask about what a clinical development program might look like, or what preclinical data would need to support an IND. But I just want to get back to what's really going to have to happen for us to take the next step. Well, from our vantage point, it's going to have to be a quantum leap in terms of manufacturing. Because if gene therapy is to address problems in high-income countries, it also actually has an incredible potential to address issues in low and middle income countries where there's actually, you can't address them any other way, because the kind of supportive care such as blood products, intravenous fluids that we can give here, they're just not going to have the infrastructure in those environments where actually a single administration of a gene therapy might be possible. To get there, we're going to have an order of magnitude, at least 1 to 2 order of magnitude improvement in our ability to manufacture quantities of gene therapy vectors. And at the same time, we're going to have to find a 1 to 2 order of magnitude reduction of cost in our ability to do that. And you say that's impossible, but there was a guy named Steve Jobs who thought differently, and so suddenly something, when I worked across the street at New York University doing my PhD, and at that time we had a computer which basically cost somewhere on the order, NIH fund is probably, the computing system's about $500,000, and right now this iPhone can do more, okay? So things can actually happen, and I think we do have to think, I think industry is going to have to think looking forward here, and we're collaborating with industry and academics to try to advance where the manufacturing will go for these products.
Good afternoon. My name is Brian Caspar, Chief Scientific Officer of AveXis, which is, as you heard from Michael, recently acquired by now Novartis. So we are a Novartis company. We work on primarily spinal muscular atrophy, this rare pediatric deadly orphan disease in which it's called Wernicke-Hoffman disease or floppy baby syndrome. These babies don't make it past their 2nd birthday. They'll never be able to sit, they'll never be able to talk, crawl, or walk, and they don't make it past their 2nd birthday. We developed a gene delivery, a one-time administration into the bloodstream that has essentially, in the phase 1 clinical trial, had 100% survival of these patients, which remains today. Many of these patients have far surpassed the age of 2, where you would expect at least 90% of the kids to either be on a tracheostomy or be dead. And we have many of our children are nearing the age of 5 years of age. Some of these kids intervened early are crawling, standing, running. Some look like just normal kids, despite having the genetic code for a lethal pediatric disease. We have grown this company. We started very similar to Kathy as an academic understanding, driving the science, moving things forward into first-in-human studies, to saying, holy cow, this is transformative, this is working, we need to be able to make this drug so that every single child, there's approximately 500 children born here in the United States that have this disease, and it's a disease, just as you heard from Peter Marks, many of these diseases are around the globe. So we have to be thinking about global regulatory strategies as well as cost of goods and manufacturing to ensure that batch number 1 acts like batch number 3, acts like batch number 303. And I think we'll hear more about this during our session here. The company has grown dramatically. We're now over 500 employees with a sole focus of moving transformative drugs to patients. The company is moving into an autism spectrum disorder called Rett syndrome, as well as our third program moving forward, a genetic form of Lou Gehrig's disease, or ALS.
So I'm Philip Gregory. I'm the Chief Scientific Officer at Bluebird. So we sort of change therapeutic modality as we move down the panel here. So as was said, Bluebird's approaches are ex vivo cellular therapies where we genetically modify cells taken out of the patient and then return those cells back to the patient. We use lentiviral vectors to do that genetic modification. These are vectors that can carry genetic payloads, corrective genes, into stem cells or T cells. And the therapies that we have been working on really fall into two classes. So severe genetic diseases, thalassemia, sickle cell disease, and cerebral adrenoleukodystrophy, a severe disease of the neural system. Those are all addressed through a stem cell transplant of these corrected modified cells. And then using essentially the same approaches, we can do CAR T-cell type cancer therapies where we use lentiviral vectors to carry redirection technologies that teach those T-cells to see specific targets on the surface of tumor cells. And so those are the sort of the types of things that we work on. Just to piggyback on something that Peter said, we've had at Bluebird a very sort of innovative culture, and we've set ourselves the task of good being never good enough for patients. And particularly on the manufacturing side, we've continued to innovate. In fact, the latest generation of our thalassemia product has a different manufacturing process, which massively improves the efficiency of transduction and the amount of vector we need to use and so on. And similarly, on the CAR T-cell front, we have a second generation already manufacturing process that we think will improve the durability of those cells. So we've continued to think about ways in which these generation 1.0 manufacturing processes need to be updated as we move to hopefully more robust outcomes, but ultimately to try and get these therapies to a broader audience.
I see you don't wear ties at Bluebird. Might have to learn something here. Lose the tie, maybe.
This is as smart as I've ever been this week.
That's great. Or AveXis, looks like. Gaurav Shah, I am the CEO of Rocket Pharma. We're actually next door here. And in some ways, we've learned a lot from everybody who's here. We're inspired by the work that's been done already by those who have gone before us. I'm happy to represent an earlier stage company. We're in the clinic in one indication, Fanconi anemia. We're preclinical in 3 other ex vivo lentiviral-based indications, and we also have an upcoming AAV indication that we'll disclose later this year. I'm also happy to represent sort of the emerging New York City biotech sphere. Alexandria Center here has been instrumental in making sure that we get together, we learn from one another, and that we can collaborate. So I had the reverse commute problem, sort of the opposite of Brian, who went from biotech to Novartis. I went from Novartis, where I worked on CAR T, to a one-person company called Rocket Pharma 3 years ago, sitting in a conference room alone for many months, trying to figure out what to do. And it allowed me, in some ways, to ask questions that I think are important. One is, how do you integrate better? Just from my prior lives, how do you integrate the thinking across collaborators, across to the agency here versus Europe? How do you integrate with patients and patient advocacy groups? That was a question that I sat and thought about a lot. Also, how do you integrate within the company? CMC, clinical, regulatory, science research, with an eye to commercialization at some point. How can we think about this early on so that we can really do it right and efficiently over the next several years? And the other thing that I thought a lot about is definition of self. Who are we? Who are we as a company? Are we a gene therapy company? Are we a rare disease company or something else? And maybe we can talk more about that a little bit later. But thanks for having me here, Michael.
Thank you very much. Kathy, one question that comes to my mind after you have embarked on local delivery to hereditary eye diseases, IV to target the liver for hemophilia. Do you now feel that you have enough platform capabilities around AAV, so the challenge and what excites you is to expand that to more diseases, or do you see in front of you a near-term opportunity to advance the technology into that gives you access to areas beyond current reach?
So I will say that for me, an important realization when we began Spark and a foundational observation that let us do it was that I believed that there were enough problems solved for AAV delivery to a few target tissues to actually allow us to do drug development in that space. So it did not mean that every single problem was solved for AAV delivery to the central nervous system, for example, but it meant that there were enough things solved that we could make a reasonable approach to a whole series of diseases that can be approached, for example, by giving AAV to the subretinal space or introducing AAV intravenously to target the liver, and so forth. So have we solved everything that we need to solve to target every disease that can be approached by delivering AAV to the liver? No. I do believe, and this echoes a theme that we've heard earlier, that good clinical results drive better process development, which expands manufacturing capability and allows you to approach diseases that you couldn't before because of manufacturing yields and the quality of the product and so forth. And then that allows more clinical results, which fuels more work in manufacturing. So I think we will continue to see an expansion of feasible targets in gene therapy.
Thank you, Peter. When it comes to regulatory harmonization, as just illustrated by the colleagues here, there are a lot of good dialogues, but still different guidances and processes. Do you foresee an opportunity to gather key regulators across the globe on, let's say, the major bottlenecks? And one thing that comes to my mind is that for some of these patients, the disease is really progressive. So having a treatment available 1-2 years earlier would change the life destination of that patient and that family. So where do you think we are in our dialogues around being able to define surrogate endpoints that may, with just some months of clinical data, allow regulators to feel that it's sufficient probability of sustained benefit given the severity of the disease to accelerate? Do you think this dialogue is going to happen, or do we need just to have more patience?
So I can say from the standpoint of dialogue with, let me start from the dialogue with international regulators is very much ongoing. And I think in the past year we've made a lot of progress because I think high-income countries, for instance, the United States and the European Union really understand that it's very challenging for a developer to have different standards in different places and to have to go in and hear different scientific advice in 2 different places. So we understand that, and we are increasingly trying to get that harmonized. We're starting to encourage people very early on in their development programs to come into us and actually to invite EMA to listen into their discussions so that EMA early on understands where things are headed. That's very helpful with dealing with this issue of surrogate endpoints, because I think one of the things that we've learned is that this is an incredibly fast-moving scientific field, and we have to constantly keep up with the developments, CRISPR-Cas9, and now CRISPR-Cas12. There's all sorts of things that keep coming down the pipeline, and we have to keep up with that. So to the extent that I think we as global regulators are involved together as these things develop, it's helpful as we come to get them towards approval. Surrogate endpoints, I think we are very comfortable in certain areas with surrogate endpoints. I can't speak for other regulatory authorities, but I think we do have a chance. Someone made a joke about globalism earlier today, and I'm going to make a joke. I mean, from a global perspective, we have a chance to be a lead as a lead regulatory organization, because people generally do, they do at least listen to what FDA has to say globally. And I think if we are out in front with where our acceptance of some of these surrogate endpoints or intermediate endpoints are, I think some will come along.
Thank you. Let's take the opportunity to open up for some questions and come back to the panelists to mix it up a bit here. On this side here.
For those of us who are not, experts in molecular genetics and gene therapy, how are you thinking about approaching polygenic diseases? And you mentioned autism, which is obviously one of them.
Brian, you want to start?
So let me just state that the first autism spectrum disorder that we're focused on is indeed a monogenetic disease with loss of expression of the MECP2 gene. So I think this field has really grown on and advanced in thinking about very defined targets, being the monogenetic diseases, and where we can establish the safety, number one, but also the dosing parameters and understanding of the design of clinical trials and the measures of clinical benefit for patients. I think the field is growing to be able to now think about how we take that knowledge and grow with all of those aspects, from clinical trial design, understanding the product, understanding the safety, understanding how to manufacture and scale, understanding how to control your product and know your product better than we did even 3 years ago. There are now tools out there that we can really think about new defined targets to go after, larger diseases like Alzheimer's disease or congestive heart failures, where there might not be a defined target, but potential therapies for the future.
Thank you.
Just to illustrate the complexity, because I'm in no way not mindful of that, what has been the difference and why has it been so difficult for other monogenic diseases like cystic fibrosis or Huntington's?
Well, I think that what it really turns out to be the case, at least for in vivo delivery of AAV, every target tissue is essentially a different problem. And part of that has to do with the human immune response, which is also a tissue-specific response. For something like cystic fibrosis, you have the additional complication that where you really need to get to is the basal respiratory epithelium, which continually turns over, gives rise to the cells above that continually turn over. And AAV is not an integrating vector, and so it will be lost from the cells at the top of the respiratory tract that are sloughed. So there are many complexities, I think, that have to be dealt with as you branch out to all the different target tissues.
I think this opportunity to expand across diseases is very tempting, but I'm personally quite fascinated about the you could stepwise approach that you alluded to, Brian. While we started in a single gene hereditary rare disease, given the tremendous benefit risk for new modalities we offer for those patients with well predicted lifespan without new treatments, to then go into common diseases but genetic subtypes where you again have removed the understanding of the target validation, because it's really about restoring a single gene function. It's just part of a more common clinical disease phenotype. I think that's really the path to go. Once you branch into polygenic diseases with environmental influence, it will be so much harder to tease apart technology shortcomings, delivery, and how much each gene play a role in various symptomology. So it certainly would be good for the field to have moved stepwise and gained confidence. Other questions?
Hello. So how do you see the IP of Cas9 and Cas12 affecting, it's great as a research tool, but now that it's becoming commercialized and it could be used in a variety of gene therapies and cell therapies, but UC Berkeley and the Broad Institute has given this license to many companies and it really has created their value. How do you see this then affecting other companies trying to implement this strategy, whether it be in cell or gene therapy?
Just one context here. I guess while we mainly currently focus on gene therapy by delivering synthetic gene, the question is more about gene editing, which is a field still, I think, need to overcome a lot of technical hurdles. It's a much younger field. So I think the IP there is different from gene therapy, but maybe Phil can share some thoughts.
Yeah, so in a former life, I was the CSO at Sangamo. So I guess on one level, you could say, well, I don't think it's our purview to sort of get wade into the heated debates over the IP around the CRISPR-Cas system. In all likelihood, like with any other very complex IP setting, there will have to be cross-licensing at some point. That seems to be the most likely prediction. And I think no company no institution, no company is interested in blocking access to patients' therapies that could be driven by these technologies. That said, of course, there are other editing technologies. So there's the ZFP platforms, there's the Talon platforms, Bluebird has its MegaTal platform, and I'm sure that there's more coming that we haven't even heard of yet. So I'm sure there's more coming in this regard. I think it's a very powerful approach. It's very complementary to the current approaches with more classical gene addition technologies, such as lentiviral vectors and AAV. And I think it's an important component of the portfolio of ways in which we have to address different types of disease. So I hope that provides at least some answer.
While we wait for additional questions, I'll continue with the pointed question, and I think I managed to get through Kathy and Peter here. Brian, I thought I'll ask you a little bit more of a talent question that can stimulate new entrepreneurs. When you made this journey from building competitive gene therapy programs in academia, starting your own company, bringing it to late-stage clinical, and then entering big pharma. If you think back, is it possible for you, with a single word in each stage, say what was the most important personal trait you had that made success here?
Boil it down to
This
one
was
word.
not prepared, so I'm
Thank you,
making it tough. But I've been on other panels with Brian, and he's just so good, so I need to up the game here.
I think tenacity is a word, and I have to throw one more word in there.
You can have one word for each challenge. Academia to biotech, biotech to registration, and then big pharma.
Tenacity all the way.
All the way.
Teamwork is essential. This is not a one person's win. It really takes a village and an army to get these things forward. It also takes a talent pool that I don't think academics really understood, and I'll admit to that. I actually don't think the public understands the complexity of drug development. And actually, I don't think even in large pharma, everyone understands the complexities of bringing forward drugs globally. That isn't to bring things down. It is just a tremendously difficult path. Not everyone knows how to get there, but when you pull together talented individuals when you take advantage of the programs that Peter Marks just mentioned, and you work through global harmonization that is occurring within regulatory aspects, you can take advantage of breakthrough therapy designation, EMA prime designations, and even Japanese Sakigake, which we've garnered for AveXis 101. So everything that Peter mentioned is actually occurring and can be taken advantage of as we pioneer new spaces. So then I think it's, let me use the final word, is integrity. On the aspect that we have to pay attention to our science, we have to pay attention to the challenges and the pitfalls as you're pioneering, and you have to maneuver in a way keeping integrity at your center core to really get things across the finish line. And I think if you take the teamwork, tenacity, and integrity, you really have created something that now you have a fighting chance.
Thank you.
So a little unusual. I just have a comment for Brian. My niece has, I'm Omar Manjwal, I'm the Chief Medical Officer of Catharsis. My niece has Rett, and I was texting her dad to tell me that to tell him that you were here, and he has a message for you, which is hurry up.
Okay.
Yeah. It's on. I think when some investigators, some 20, 30, 40 years ago, started dreaming about genetic therapies, they had in mind the prospect of a definitive treatment that could be curative, administered only once, and that might decrease the cost of medicine. These first therapies have cost between $500,000 to $1 million an injection, and for some people have projected this image that this is a boutique therapy for a few people in a few rich countries. And I'm curious to know what each member of the panel actually thinks about where things might go with respect to the cost of these therapies, what they foresee for the next 5 or 10 years.
This is Michel Sutherland that works on low-cost therapy called CAR-T, by the way. I couldn't resist joking a little bit. But, a general comment was that I have, because we thought we'll keep the panel focusing more on the promise of these treatments that then end up in a price discussion, is that in general, I think what we want to see is that treatments are rewarded for the value they provide to patients and society. And of course, we want to sustain innovations. And some of these innovations take decades with uncertain outcome. Investment in gene therapy has probably been over 3 decades. So it's a thoughtful balance of how you reward value to patients, to the healthcare system, and sustain innovation. And of course, that doesn't mean that the price should be equal in each region of the world, and it often is not. And I think everyone is eager to make sure that patients in the part of the world where they cannot afford healthcare, as in the developed world, can have access. And I think, Peter, you spoke about the improvement in manufacturing that is important to make that happen. If you want to add some words, but I just...
We don't approve things based on pricing. That said, I will tell you that we live in a large ecosystem, and gene therapy, we are not the only place that gene therapy is being developed. And I think that others around the globe, in some very, very populous places, perhaps in Asia, realize the benefit of this. And eventually there will be a competition here will drive the cost down. And admittedly, it's quite expensive. I'm a hematologist, oncologist by training. I treated hemophilia for quite a number of years. Yes, the first hemophilia therapy is probably going to have sticker shock, but if you know what actually the cost is per year of treating hemophilia now, it's not all that crazy, perhaps, because right now the cost of a person with hemophilia now uses, basically if you don't have factor 8 or factor 9 in your blood, you basically take prophylactic infusions with a factor concentrate that's quite expensive, can be between $100,000 to $200,000 a year, plus all the other ancillary costs. So again, these things, it's a balance. But I think ultimately, it's going to be a competition as more of these come in, and as we develop economies of scale for manufacturing and improvements, which I think everyone's spoken about here, is as we have more experience, it's incremental building of knowledge that will build on top of itself to allow lower cost of manufacture. And then I think, I don't know you're going to go to the dime store to buy the stuff, but I think we'll see an improvement.
Thank you. And clearly, everyone involved in this type of demanding R&D would like to see that access is overcome to allow these transformative treatments, whether CAR-T or gene therapy for rare disease, to be available. Let me just shift a little bit and ask Phil to address something different. At Bluebird, I've been impressed with, you put a lot of emphasis on the culture to drive creativity and innovation. And what is it in the Bluebird nest that allow you, as you have grown from a little bit of a boutique company to quite a large biotech, to maintain that feel of everyone matters in making a difference for patients, as we heard from a dad texting to a child?
Sure. So well, we actually do call it the nest. So that's, it really isn't a joke. That's the name of home base. I think you can really divide our culture into two pieces, and they sort of have names. One is True Blue, and that's really our patient focus, and that really is the center of Bluebird's culture. It's we live that patient focus in a way that no other place that I've worked at has achieved that same sort of patient-centric view. So literally in every leadership team meeting, someone will raise the question as to what the right decision is for patients, right? And that becomes our true north, that's our guiding principle. And that filters down through everything that we do. And we bring patients, patient advocacy organizations, caregivers, that are working with patients and have the diseases that we currently treat. We bring them into the company on a regular basis so that we can understand the diseases they have, understand the impact that the disease have on their lives. And that, I can tell you, is incredibly motivating as somebody on the bench research side. It gets you out of bed in the morning knowing that if we can get this past the next goalpost, we might actually have a chance at changing the course of disease for these patients. So we really live that patient centricity in a way that's different. But we've also realized that to do that, we also have to think about not only our patients, but our people. And that's sort of the blue mojo side of what we talk about. So, and that's trying to think about how we can do everything we can as an organization to support the workforce, to support the employees, to be able to deliver on these astonishing therapies and try and do something truly transformational. And so it's really that combination of putting the patient first, but realizing that we're only going to get there through looking after our people.
Thank you. As we speak about the patient centricity as a complement to advancing new modalities, so Gaurav, as you've been thinking a lot as a physician and scientist on advancing these therapies, how do you balance between advancing a new gene therapy that may address gene deficiency in some organs, but possibly not all organs, and not diminish all symptoms of a patient, meaning you may need to have a further refinement of an additional vector, versus moving with a vector fit for purpose for a certain organ into new diseases? So how do you see that balance of full remission for a patient, continuing with that patient focus, versus the opportunity to make an immediate impact for another patient group?
Yeah, thank you. I think that, again, being in an early stage company, it's allowed time for thought and reflection, right? And one of the things at Novartis that I never really got to do was engage a lot with patients and physicians who are out there in the real world. It's something that now I get to do a lot more. So we're sponsoring some academic trials where we're asking these very questions. Are we treating just to get an approval, or are we treating the whole patient over the course of their life? And Fanconi anemia is a perfect example of the differentiation there. This is a disorder of DNA repair in which every cell in the body has difficulty withstanding the onslaught of oxidative and other stresses because of a DNA repair issue. Now, the main manifestation of Fanconi anemia and the cause of early mortality is hematologic. Patients often pass away from bone marrow failure and then otherwise MDS or AML if they make it into their teenage years and 20s. So while a lentiviral ex vivo bone marrow stem cell approach has the potential to cure the hematologic manifestations of Fanconi, those who do survive the hematologic onslaught, 80% of these patients die of head and neck tumors that also arise from the same DNA repair issue but is not addressed by ex vivo lenti. So now we're talking to the Fanconi Anemia Research Foundation and the community around the world that sees these patients, and they're asking, well, okay, you're going to cure potentially the bone marrow problem, but what about the solid tumor? What about the head and neck cancer? And this has, again, led us to think hard about, are we a gene therapy company? Are we a platform company? Or are we a rare disease company? And I think there's increasing value as a development organization, rather than company, as a development organization to move toward the disease-oriented approach. I think there's a lot of value that should take some light in coming years.
Thank you. Any more questions?
This is a question. I'll yell it out. This is a question about vector technology. On the one hand, I'm stunned by how far we've gotten with AAV and Lenti, with all the applications you guys talked about in the introduction and things that Michelle and others have done. But on the other hand, the vectors that we're currently using have small payloads. Their tropism is difficult to control, and there are sometimes problems with immunogenicity. And of course, taking cells out of the body, transducing them, and putting them back is difficult to scale. So what do you guys see for vector technology improvements or other delivery modes going forward?
Okay, well, you're preaching to the choir with that question, but I will say that one thing I've learned over many years is not to make the perfect the enemy of the good. We do have gene delivery vehicles that we can use now for drug development. There will always be better things coming down the pipeline. Not all of those things will deliver on their promise, but we continually scan the environment for novel gene delivery vehicles that may solve some of the problems that you alluded to in your question. So again, I think, success breeds success. There will be second generation and third generation gene delivery vehicles.
So maybe I'll add on to that with a really good question, and maybe I can pull in Brian here on this one. One challenge for the future is that we don't know fully the sustainability of the transgene provided to patients. We are optimistic that they may last years or decades, but we don't know how many of those. So how do you see the field of retreatment developing, whether it will be further engineered vectors where you remove immunogenic epitopes, or you think we will move away from viral capsids?
So, Michael, it's a great question. It's something that all of us sitting on this stage think about, and we would be remiss, even though academically and theoretically we have never seen anything that would suggest that these things are turning off over time, and that the durability is there. Okay, we're not 40 years out. We're not even in 30 years out. So I think the field is starting to think holistically about this, where many of us are paying attention to this. There are thoughts of engineering vectors to get around the immune responses, to allow for redosing. There's better and better immunological products to transiently suppress the immune system, where there's, I think, already some exciting advances of thinking about plasmapheresis, for example, combined potentially with immunosuppressive regimens that would allow for redosing to occur. And then I think you start thinking about, routes of administration. For what we've been working on thinking about SMA, for example, where we're treating patients systemically, perhaps redosing might only have to go back to the CNS only, and an intrathecal route of administration could be afforded there. So having routes of administration combined with thinking about immunosuppression, and then as Kathy was mentioning, we are all scanning the field for improvements. And that sometimes can happen overnight and sometimes happens over years.
Please, a question in the center there. We have a mic, or you can shout.
Hopefully next. I'm going to shout. So it's a more scientific question. Of course, modifying the genetic code of the patient cells and then putting back the cells in the patients is more complicated than small molecules, right? So what are the challenges with the quality control, avoiding like toxicity or adverse effects, and what do you think about that, and what are your thoughts about it?
Peter, maybe you want to start.
So I'm going to try to be, I don't know how much time we have, we may be running short, but I'll just try to say that this is a quantum difference between small molecules we can characterize extremely well. You can use mass spec, high performance liquid chromatography, you know what's there. These things are complicated, and I think it's a compliment to all the companies who have been involved that they've done tremendous process engineering to develop critical quality attributes on how to manufacture these things. Is it perfect? They'll tell you it's not yet, and there's room for improvement, but they've done incredible work, I mean, making products where they've had to come up with various controls along the way, from everything from making sure that the DNA from vector production, doesn't come along with all sorts of things. So very complicated, and that kind of is what's helping to explain why. Again, I'm not the cost guy, but it does explain some of the complexity in manufacturing that goes into this, and the investment of having to re-engineer this. This is part of what the pioneers are having to do. It's not that you can go to there's not like a cookbook that you can go to and say this is how you do it. So quality systems are being re-engineered here. And at some point, I think it'll settle down, and that will also help bring costs down, because there probably will get to be more like a cookbook, and there'll be a cadre of people, professionals, who are used to doing this, who will go between companies probably, like everything does, to the highest bidder, and that will probably make things. But right now, it is a challenge, and that's part of what it's about.
Yeah, I think the one way to think about it is that when you think about small molecules, drugging a kinase, for example, what you worry about is drugging another kinase you didn't want to drug, right? So when we think about making modifications to DNA, essentially our target substrate is DNA. So our worry is, did we do something unintended to that same DNA when we were doing the good thing? And that's the framework through which all the assays that we've built, both on the lentiviral vector gene therapy side as well as the gene editing side, are all focused on what are the unintended consequences of that modification. Is it an off-target? Is it an insertion in a gene you didn't want to insert in? And then building all the technologies that allow you to see that in incredibly fine resolution and characterize your drug products so you understand that risk. The risk won't go away. Your processes can be improved to limit the risk, but understanding that risk is really important.
Great discussion here, and I appreciate the breadth of dialogues from pricing to patient centricity to relatives here. So I wanted to make sure we didn't miss out in listening to the future to come from our esteemed panel here. So close your eyes for a few moments. We are back here, late October. It's a warmer New York day, 2023. And science is moving rapidly. There is political consensus in society to support healthcare for everyone. And what would be the most exciting thing? We'll start with Gaurav and move forwards, happening in the pipelines of companies like yours that you would be proudly speaking about.
Yeah, I think, to be brief, moving from bench to bedside to therapies being available for patients around the world, shortening that time span is going to be very important. And I think getting an idea, saying there's a disease that we want to cure. We have a vector or a gene therapy modality that's appropriate, moving into clinic, moving into commercialization. That path might take 10 years right now. I think it'll be really short in 5 years as we consolidate a lot of the thinking here.
So I finally get to answer Michelle's question, which is that I think in the next 5 years, what you're going to see is incredible innovation on the manufacturing of both the drug products and the vectors that will massively change the cost of goods of these drugs, and will actually make it much more likely we can move these outside of rather privileged societies like our own, and hopefully to underprivileged, and get these drugs to where they're needed.
I think I'll hold myself to one word. I think we can change this to the reality of gene therapy. We will be seeing successes. We will have the infrastructure available to, and 5 years goes by very quickly, as we all know. So I think we'll have more than just a handful of successes.
I think from the regulatory perspective, we'll probably be in a place where there will be enough successes out there that we will have learned enough that we'll be trying to get towards finding ways to expedite therapies by having enough experience, for instance, with vectors, that we may be able to bypass some of the traditional toxicology and other things that we're currently having to do. So I think it'll be finding even further expeditious regulatory pathways.
So, Peter, 2023, you think how many drugs of the character of gene therapy will be approved? And number two, do you think you will be seeing clinical studies with gene editing, any late-stage studies, or like gene therapy, it is a long journey to go? Not gene therapy today, but 3 decades ago.
So the first one I'm going to punt and just quote MIT. MIT has predicted that in 5 years from now there'll be 30 to 40 gene therapies approved. That's not from the FDA. That's from MIT. And I actually think that gene editing is moving. Gene editing technology. I'm not saying a particular gene editing technology. There are a variety of gene editing technologies in development, but I think gene editing technology will be in late stage development, or actually. I would hazard to even go out on a limb and say, is it possible? It's possible that you could even see an approved product based on a gene editing technology. So, yeah.
And we're still talking about monogenetic rare disease or heart failure or dementia.
I think listen, aim high. You could imagine that there are polygenic diseases that could be addressed by dealing with one gene, for instance. For instance, atherosclerosis, you could imagine that if someone could reduce your cholesterol by interrupting a gene and bringing it down 50%, you would probably decrease mortality very, very significantly from atherosclerosis causing heart disease and stroke. And so I think that it's possible that somebody might, that's how I think people will first deal with polygenic diseases, is they'll find one target that they can hit that can bring things down. The problem is to get there. Listen, if you had it today and it was effective, you couldn't make it in sufficient quantity for the demand. So it's going to take the field to mature to get there. And I think it's a great goal for industry, academics, and government to work together to get there, because it really has a chance to transform global health.
Well, I don't have much to add, but I'll just say that I think this has been a remarkable panel, and I think if you look across the companies represented here in the next few years, I believe that we will see gene therapies for hemophilia, for spinal muscular atrophy, for thalassemia and sickle cell disease, all cross the finish line. And I believe that from there, as you alluded to, we will be able to move to more complex acquired disorders with genetic strategies. I believe that ongoing work in genome sequencing, wide numbers of the population, will allow us to apply these in the most intelligent fashion, will allow us to stratify populations in a way that we can get genetic strategies to work for complex acquired disorders as well.
Thank you very much. I hope you've enjoyed this journey through gene therapy, and having had Q&A and listening to the insight from some of the most leading transformative discoverers and developers that are here. And thank you, Peter, for generously sharing personal and FDA thinking in this space. You know, as we go from having had a more incremental impact over some time on human health to be able to think about curing, it's so fascinating. That word with just a few letters in it can mean so much, and what was beyond the reach of our imagination is now within the reach of every year another impactful clinical advance. So stay close to this exciting field, and welcome to further dialogues as we conclude in the time off in breaks, or just connect to any of us here offline. Thank you very much.